Hello everybody, we're back. We're going to continue on now here in Chapter 3 and talk about the plasma membrane, which as I mentioned in the first lecture for Chapter 3 is the covering around the surface of the cell. And I also mentioned that maybe surprisingly, it is a part of the cell that we talk about a lot in anatomy and physiology. And so we're going to spend some more time here in Chapter 3 introducing you to some basic information about it. All right, so your plasma membrane, what is it actually built from? And incidentally, let me point out some terminology here. Plasma membrane, sometimes people will refer to it as the cell membrane, a little bit more casual lingo. Now, cells have other membranes on the inside of the cell. There are similar membranes that cover some of the organelles, those little specialized compartments in there. So technically those are cell membranes also, but sometimes those are called intracellular membranes because they're inside the cell itself, covering features on the inside of the cell. Also, it's not unusual to hear people call the plasma membrane the cytoplasmic membrane because it is the membrane surrounding plasm, all of the contents inside of a cell between this membrane and the nucleus. All right, what is it built from? It is a double layer, or that's sometimes called a bilayer, or you can think of it as two sheets, you know, almost like two pieces of bread, almost like a sandwich that's put together. And that double layer is built from lipids and proteins. The lipids that are used to make it up are called phospholipids. We heard a little bit about those in Chapter 2. The plasma membrane is often referred to as a fluid mosaic. So if you've heard of mosaic before, like a mosaic, that means that the tiles are used in a pattern that is not, you can have different colors, different patterns scattered around somewhat randomly within the pattern. And plasma membrane is kind of like that too. These, you can have different membrane and you know, that have different functions and different roles, and they're scattered around. It's not, they're not in any type of uniform pattern there in that part of the cell. And what does the fluid part of the name mean? Now, you're used to thinking about fluids being inside a cell and outside the cell, but the membrane itself, that barrier, it's not like a piece of saran wrap or a concrete barrier or something. It actually flows around the surface, which is can be a little bit difficult to visualize at first. So that's what's meant by fluid mosaic. Plasma membrane plays a very dynamic role in cellular activity. Dynamic means active. So again, it's not just a covering. It plays very important roles in determining what goes inside the cell, what comes back out. The plasma membrane has to receive signals from other cells and glands in the body, and so because of that, we think of it as being a very dynamic type structure. Plasma membrane separates intracellular fluid from extracellular fluid, or ECF. So if this is a cell, of course you have fluids, watery-based fluids inside the cell. Those are referred to as intracellular fluid, or ICF. And then out here, fluids outside the cell are called extracellular fluids. So that prefix extra can mean outside, you know, like the word extraordinary, extraordinary, outside of the ordinary. So that's how that prefix is being used here as well. You have lots of extracellular fluids in your body. Anywhere where you have fluid and it's not inside a cell, it's not part of the internal structures of a cell, that's considered to be extracellular fluid. So you've got blood, your tears, your the fluid that surrounds your brain and your spinal cord, those serous fluids in your peritoneal cavity and pleural cavity, pericardial cavities. All of those are examples of extracellular fluids. Now, more specifically, you also have extracellular fluids, of course, that surround your cells and your tissues. So your tissues are all built from cells, and those cells, you know, kind of like if you think about bricks being cells and how you've got mortar between the bricks, you've got filler spaces between cells and your tissues, and those filler spaces are going to contain fluid that is a type of extracellular fluid, but it's not blood, it's not serous fluid, it has its own name. In this case, it's called interstitial fluid. Interstitial fluid equals tissue fluid. For example, like if you think about the wall of your heart, for example, has lots and lots of muscular tissue in it. There are lots of muscle cells in there that contract to allow your heart to beat. And there are fluid-filled spaces that surround each of those muscle cells. And that fluid is a type of interstitial or tissue fluid in that location. All right, how about these phospholipids, these special lipids that make up our cell membranes, along with the proteins that are there as well. Talked a little bit about these in Chapter 2, but let's review a little bit here. Phospholipids are special lipids that have two parts. So you've got a head part over here that is water-loving or hydrophilic. And then you've got these two fatty acid tails over here that are fatty, as their name implies, and that means they are hydrophobic. They repel water. They don't like water. Okay, so that the hydrophilic portion of the molecule, the reason it's called a phospholipid is because you do have a phosphorus atom that is part of a phosphate group. All a phosphate is a phosphorus surrounded by oxygens. So those heads on these phospholipids always have that in there among some other atoms that are part of the structure. These are also called polar heads sometimes. And all polar means is you've got a molecule or a portion of a molecule where it's positively charged in one location, negatively charged over on the other side. Kind of like a battery. You have poles on a battery, a positive pole and a negative pole, like on a car battery. Same thing here, a polar molecule or a polar portion of a molecule like a phospholipid has a positively charged area and a negatively charged area. And you can actually see that if you look closely at our little phospholipid. The head there has a positive charge in one spot, negative charge in another. Key thing for us to remember, things that are polar like that, things that have charges, positive or negative charges, are going to be hydrophilic. Which means they love water. So the head portions of these phospholipids love water. Those fatty acid tails hate water. Hydrophobic substances like other hydrophobic substances, which means those fatty acid tails, they like to get together and interact with each other. So here's a chunk of a phospholipid bilayer, two sheets of phospholipids that are getting together and making a cell membrane. And so if you notice, you got one sheet over here, you got one sheet over here, and all of those heads, those hydrophilic heads, they like each other. Just like hydrophobic molecules, like other hydrophobic molecules, hydrophilic molecules like each other, and they also like water. So those hydrophilic heads, they all interact with each other. The hydrophilic heads over here on this sheet, they're all interacting with each other. And then those fatty acid tails, they get together and intermingle together because hydrophobic likes hydrophobic. So because of this, these phospholipids, because of this structure that they have, they are natural at forming these lipid bilayers where you have these two sheets that get together. You can almost think about those, the heads of the phospholipids as forming like the bread of a sandwich, and then the fatty acid tails are forming the peanut butter on the inside. And so this makes a very nice barrier. Those fatty acid tails there intermingling with each other, that forms a hydrophobic barrier between the inside and the outside of a cell. The reason for that is most of your solutes, you got solutes inside the cell, you got solutes outside the cell, and most of those are hydrophilic. So they don't like those fatty acid tails. So most solutes cannot just freely move across the cell membrane because they're not going to be able to get in there and intermingle with those fatty acid tails long enough to be able to do that. All right, here's another depiction of a phospholipid bilayer plasma membrane from your textbook. This one's a little bit more complicated. So in a light blue there you can see, or light purple, you can see all the phospholipids there forming the bilayer. And then these larger purple blobs you have on here, those are all different types of membrane proteins. They're also referred to here as integral proteins, most of them. The ones that are actually sitting in the membrane are integral proteins. The ones that are just kind of attached to one side or the other are sometimes called peripheral proteins because they're on the periphery, the edges of the membrane, rather than being embedded within it. You can see you've got many different types there. Many of them are involved in transporting substances across the membrane. So we just said that most of the solutes, things like sugars and amino acids and proteins and so forth, those types of things are hydrophilic, so they're not going to be able to easily get across the cell membrane. So what cells do, when they do have something that they need to deliberately move across, they use these membrane proteins very often to accomplish that. And we're going to see more about that. We're going to have a separate lecture video clip about transporting things across the plasma membrane. Some of those proteins function as receptors for receiving signals from other cells or glands. So if we've got a cell here, you got some of these membrane proteins sitting all over the surface of the cell, and many of those are receptors for chemicals like, let's say, hormones. Like, let's say this is a hormone. See, the little X there is a hormone molecule, and it comes diffusing along through the extracellular fluid. And if this is a receptor for that hormone, it will very strongly attach to that receptor. And that's often like a switch gets flipped inside the cell that is telling the cell it has to make some sort of response. It either has to do something it wasn't doing before or stop doing something that it has been doing. So cells and glands in your body send lots of chemical signals to each other. And then finally, I'll back up over here, attachment to the cytoskeleton and extracellular matrix. Let's back up over here and take another look at this diagram. So if you notice, you've got on the inside of the cell here all these little protein threads or filaments. Filament is another term for thread. Those are built from proteins, and they make up what's known as the cytoskeleton. It's like a protein meshwork inside the cell that helps prevent gives the cell some support. It helps prevent it from being too easily squished and compressed, and also it helps maintain the shape of the cell. And so those little threads can be attached up here to some of these membrane proteins to help anchor the cytoskeleton to the cell membrane. And then also some of these membrane proteins will be attached out here in the extracellular space out here, the spaces surrounding the cell. You have substances out there as well, large molecules that membrane proteins attach to help anchor that cell in place where it's supposed to be. All this area outside, the filler spaces around cells, is often referred to as extracellular matrix. So yes, you have fluid there, you have the interstitial tissue fluids, and then you've also got other substances in there, like many large types of proteins. We'll learn about those in Unit 2, and that help fill in those spaces. And so membrane proteins are anchored to some of those substances there to help hold them in place. All right, what else do we have here on our cell membrane? All right, these orange molecules you see scattered around the membrane, those are cholesterol molecules. Cholesterol is actually a type of lipid. kind of looks like a bunch of orange stop signs. They're linked together. That's because if you look at the chemical structure of cholesterol, it's mainly built from carbons and hydrogens that are linked together in these ring-shaped structures, and they're very hydrophobic. So you'll notice they're embedded down in there with the fatty acid tails because they like those fatty acids. They're also hydrophobic. They don't like the intracellular and the extracellular fluids. We're used to thinking about cholesterol as being something we need to avoid. It's bad. But the reality is your cells or your body makes its own cholesterol, and in addition to what we take in from our diet. And those cholesterol molecules are in all of your cell membranes, and if they weren't there, your cell membranes would not have their correct structure and function. It's just that we have to control the levels of cholesterol we have in the body. We don't want it's one of those homeostasis aspects. We can't have too much and we can't have too little. Last thing here to point out on the cell membrane, all this green stuff that's depicted sticking up from the surface, those are carbohydrates. So you do have carbohydrates. Remember, carbohydrates, Bert and Ernie, my dachshund's barking again. Carbohydrates are chains of monosaccharides, sugars that are linked together. And so you'll have those attached to membrane proteins, you'll have those attached to membrane lipids. And when you have sugars attached to a lipid, carbohydrates attached to a lipid, those are called glycolipids. Glyco sounds like glucose, it's referring to sugars. If those carbohydrates are attached to membrane proteins, they're called glycoproteins. So you have sugars attached to a protein. All right, so the next lecture we're going to talk more about how do substances get across the plasma membrane. The plasma membrane is a barrier, so it needs to keep things out of the cell that are not supposed to come inside, and also you have to allow things to leave the cell that need to leave. And so how does that happen? And we'll talk about that in the third lecture clip for Chapter 3.